Hello, I'm Joisha.
I'm Emily Williams,
And I'm Deidre.
and our project is the improved extraction of tocopherol from soybean oil deodorizer distillate and synthesis of Chroman derivatives. So just a little bit of background. Tocopherols are an antioxidant found in vitamin E. There are 4 types, alpha, beta, gamma, and delta, and they are very important to humans and are known to have cancer-preventing properties. Unfortunately, they are currently extracted using harsh chemicals in unsafe conditions. So the goals for our experiment are to extract tocopherol from soybean oil deodorizer distillate, which is a leftover from the refining process of soybean oil, synthesize γ-Trolox in the lab, which can then be used to synthesize γ-tocopherol, and find a more efficient way to extract tocopherol from SODD using naturally occurring hydrogen bond donors.
So currently done methods of extraction include molecular distillation, and though they give out high purity and good quality of tocopherols, they have harsh operating conditions like high temperature and vacuum. So for our experiment, we're using Deep Eutectic Solvents as our method of extraction. Deep Eutectic solvents are basically combinations of quaternary ammonium salts and hydrogen bond donors, and they received their attention due to their non-toxicity and them being biodegradable. And so for our experiments, we're using 2 literature-based hydrogen bond donors and hydrogen bond acceptors, and 2 new phenol-based, naturally occurring phenol-based hydrogen bond donors.
And the reason why we are using these phenol-based hydrogen bond donors is because of their polar content, which makes it easier for them to bond with the free radicals in SODD as well as yield higher concentrations of tocopherols.
And so our extraction is Vortex Assisted. And so when we combine the DESs in the SOD, we expect, and Vortex centrifuge them, we expect multi-layer extraction. And so when the DESs combine with the tocopherol and extract them from the SOD layer, the N-hexane functions as an anti-solvent extraction. So it breaks the bonds between DESs and helps us yield tocopherols. And so we expect high amounts of tocopherols to be yielded from our naturally occurring HBDs.
And so for comparison with the tocopherols, we synthesize γ-Trolox, which is a compound similar to the naturally occurring tocopherols, though we did have to restart the synthesis because we did not get the results we expected.
And you can see this down here in results. We have a hydrogen NMR graph of our synthesized Trolox. And we did have we had 1 missing peak and 1 extra peak. So that affected our molecule, obviously. And you can see what we were supposed to get versus what we believe we got. And you can see there's a random fluorine molecule here, which is not what we needed. So we're going to have to restart that synthesis. And then for analysis and categorization of tocopherols, we'll be using HPLC to analyze the tocopherols, and we'll be using UV to characterize the tocopherols and synthesize Trolox.
So we use DVPH assay paired with UV to test for our antioxidant activities, both in only tocopherols, and to compare their antioxidant activity to the Trolox.
And so for our quantitative study, we will be using that DPPH test to see the antioxidant activities of each DES to see which one had the higher concentration. And then for our kinetic study, I will be doing that in the process of making the aldehyde, which I will split into 3 samples and then change the amount of time each will sit to form our Trolox methyl ester. And below it, you can see the chart and the graph of what we expect to see from each study.
And then lastly, we of course would like to thank the ACT program, Dr. Wicker, Dr. Foss, Dr. Shug, Abiud Fatilo, and Ryan Madigan for helping us with our experiment and encouraging us along the way.
Thank you.
Thank you.